Welcome to Developmental Biology. Today, we are going to explore one of the most magical and critical moments in the creation of life, gastrulation. We will look at how a simple, featureless ball of cells transforms into a complex, three-layered embryo, focusing on two classic models, the frog and the chick. Understanding this process is essential as it lays the foundation for the entire body plan and helps us understand how congenital defects occur. Let us begin with the frog. Imagine holding a soft, hollow rubber ball. Before gastrulation, the frog embryo is exactly like this, a hollow sphere of cells known as a blastula, simply floating in the water. But to build a tadpole, this single layer of cells must be completely reorganized. Gastrulation is the highly coordinated dance that transforms this simple ball into a complex, three-layered structure consisting of the ectoderm, mesoderm, and endoderm. This is the defining moment when the embryo establishes its fundamental body plan, deciding exactly where the head, tail, back, and belly will eventually be located. Now that we understand what the blastula is, let us look at how it actually folds inward. The process kicks off at a specific region called the gray crescent, where a small indentation forms, known as the blastopore. From here, two massive cellular movements occur simultaneously. First, cells on the top half, or animal pole, rapidly divide and spread out to cover the entire embryo. This is called epiboly. At the exact same time, cells at the edge of the blastopore roll over the lip and migrate inside. This inward rolling, called involution, hollows out a brand new internal cavity called the archenteron, which will eventually become the primitive gut of the frog. These massive cellular migrations are not just random. They follow physical laws. To understand the mechanics of gastrulation, developmental biologists measure the velocity of these cell movements. We can model the distance covered by the migrating ectoderm during epiboly using a fundamental kinematic equation, D equals V times T. Conceptually, this comes from basic physics, where distance is the product of speed and time. Here, D is the total distance the tissue has migrated in micrometers. The variable V is the average velocity of the cells in micrometers per hour, and T is the time elapsed in hours. If the velocity increases, perhaps due to warmer pond water, the distance covered in that same amount of time will also increase, speeding up development. Before we move on, let us see this with a concrete example. During frog gastrulation, the animal pole cells must migrate downward to cover the yolk plug. Suppose we observe that the total distance the cells need to cover is 1,200 micrometers, and they are moving at a constant velocity of 60 micrometers per hour. We want to find out exactly how long this developmental phase will take. We start by choosing our kinematic formula because it directly relates distance, rate, and time. We plug in 1,200 for D and 60 for V. By dividing 1,200 by 60, we find that T equals 20. Physically, this means the embryo will spend exactly 20 hours in this critical phase of epiboly before the ectoderm completely envelops it. Now that we have used this formula to solve a specific case, let us understand the general mathematical reasoning behind isolating the time variable. In biology, we often know the required distance a tissue must travel and can observe its speed under a microscope, making time the unknown factor we want to predict. We start with our fundamental equation, D equals V times T. To make this useful for finding time across any embryo, we need to isolate T. We do this by dividing both sides of the equation by the velocity, v. This algebraic step is crucial because it shifts our mathematical focus from measuring how far the cells have gone to predicting exactly how long the developmental window will remain open. Continuing from our previous step, we cancel the velocity term on the right side of the equation. This leaves the time variable, t, completely isolated. Our final rearranged formula is t equals d divided by v. This simple but powerful fraction allows us to plug in our known biological values directly. By substituting 1200 for the distance and 60 for the velocity, we perform the final division to get 20 hours. The intuition here is that time is just the total journey divided by the pace of the travelers. This proves that as long as we can measure the physical dimensions of the embryo and the speed of its cells, we can perfectly predict the timing of its developmental milestones.
Let us bring all these concepts together and look at what is actually happening inside the embryo. Here we see a cross-section of the frog during gastrulation. Notice the blue outer layer? This is the ectoderm. The arrows pointing downward along the surface represent epiboly, where these cells are spreading out to cover the embryo. On the right side, you can see the blastopore, the slit where the cells are rolling inward. This inward rolling is involution. As they move inside, they create that large yellow space in the middle, which is the archenteron, or primitive gut. You can also clearly see the three distinct layers forming: the blue ectoderm on the outside, the red mesoderm in the middle, and the yellow endoderm on the inside. This diagram perfectly captures the physical forces building the living creature. We have seen how a hollow ball folds, but now let us imagine a completely different scenario. Think of baking a pizza. The early chick embryo is not a hollow ball. It is a flat disc of cells called the blastoderm, sitting on top of a massive, unyielding ball of yolk. Because this yolk is so huge and solid, the cells simply cannot push inward like they do in the frog. Instead, they must perform a different kind of dance. The blastoderm is split into an upper layer, the epiblast, and a lower layer, the hypoblast. This flat disc model is a brilliant evolutionary adaptation that allows birds, reptiles, and even mammals to develop their complex layers on a flat plane before eventually folding up into a three-dimensional animal. Since the chick embryo cannot fold inward, how does it get cells inside? The defining feature of this process is the formation of the primitive streak. Imagine drawing a line down the center of your pizza dough. The primitive streak is a visible thickening along the midline of the epiblast. Cells from the outer edges slowly march toward this center line, fall into the crack, and spread out underneath. This dropping down is called ingression. The first cells to drop through displace the lower hypoblast layer to become the endoderm. The cells that drop in next spread out in the middle to form the mesoderm. Finally, the cells that never drop in and remain on the surface become the ectoderm. This streak is the primary organizer of the embryo's body axes. At the very tip of this primitive streak lies a fascinating and specialized structure called Henson's node. Think of it as the master conductor of the embryo. Once the primitive streak has fully extended across the flat disc, Henson's node does something remarkable. It shifts into reverse. It begins to move backward, or regress, toward the tail end of the embryo. As it travels backward, it leaves a trail of specialized tissue in its wake, much like a paving machine laying down asphalt. This trail is the notochord, which is the direct precursor to the spinal cord and surrounding vertebrae.
Tracking the movement of Henson's node is absolutely critical for biologists because it tells us exactly when and where the central nervous system is being constructed. Because the regression of Henson's node is so vital for spinal development, we can mathematically model its position over time. The formula is P of t equals L max minus the quantity V sub r times t. Conceptually, this equation states that the current position of the node is simply its starting maximum length minus the distance it has traveled backward. Here, P of t is the position of the node from the tail end in millimeters. L max represents the maximum length the streak reached before regression began. The variable V sub r is the velocity of regression in millimeters per hour, and t is the elapsed time. If the regression velocity increases, the subtracted term becomes larger, meaning the position, P of t, decreases much faster, and the node reaches the tail end sooner. Let us apply this formula to a developing chick embryo. We are given that the primitive streak reaches a maximum length of 2.5 millimeters. Once regression begins, the node moves backward at a constant velocity of 0.1 millimeters per hour. We are asked to find the exact position of Henson's node after 10 hours of regression. We choose our regression formula because it accounts for a starting position and a backward rate of change. We substitute 2.5 for L max, 0.1 for the velocity, and 10 for the time. First, we multiply 0.1 by 10 to get 1.0. Then, we subtract 1.0 from 2.5. The final answer is 1.5 millimeters. Physically, this means the node is now 1.5 millimeters from the tail, having laid down exactly 1.0 millimeter of spinal cord precursor. So, we have successfully used this formula, but let us break down the mathematical reasoning behind why it works. This equation is essentially a geometric subtraction problem. We are trying to find a remaining length by subtracting the distance traveled from the total initial length. The first logical step is to figure out exactly how far the node has moved backward. We do this by multiplying the regression velocity by the time elapsed, which is the V sub r portion of the equation. By plugging in our specific values of 0.1 millimeters per hour and 10 hours, we calculate an intermediate result of 1.0 millimeter. This tells us the absolute length of the notochord tissue that has been laid down so far. Now for the final step of our logical derivation. We know the node started at the very top of the primitive streak, which we defined as L max, or 2.5 millimeters from the tail. We also just calculated that it traveled 1.0 millimeters backward. To find its current spatial coordinate, we simply subtract that traveled distance from the original maximum length. 2.5 minus 1.0 gives us our final position of 1.5 millimeters. The intuition here is straightforward. As time moves forward, the node moves backward, shrinking the distance to the tail. This mathematical model perfectly mirrors the biological reality, ensuring biologists can verify that the central nervous system is forming at the correct spatial coordinates. Let us visualize this flat disc model with a 3D schematic of chick gastrulation. Here, you are looking at the blastoderm. Notice how different this is from the frog's spherical embryo. The top blue layer is the epiblast, which will become the ectoderm. Right down the middle, you see the deep groove of the primitive streak, with Henson's node sitting at the far end. Look closely at the red arrows. They represent the cells migrating toward the center and dropping down through the streak. This is the process of ingression. As they drop, they spread out to form the red mesoderm layer in the middle and the yellow endoderm layer at the bottom. This diagram beautifully illustrates how a flat sheet of cells solves the physical problem of a massive yolk to create three distinct layers. We have covered a lot of ground today, so let us review the most important ideas. First, we saw that frog gastrulation transforms a hollow ball of cells through the sweeping movements of epiboly and the inward rolling of involution. Second, we learned that the chick embryo, sitting on a massive yolk, must develop on a flat disc. It achieves this by forming a primitive streak, where cells drop down through ingression. Despite these very different physical mechanics, both strategies achieve the exact same goal: establishing the three primary germ layers, the ectoderm, mesoderm, and endoderm. Finally, we saw how mathematical models allow us to precisely track these movements. You now have a solid understanding of how complex life begins to take shape.